This C4 pathway is also known as dicarboxylic acid cycle, Hatch and Slack cycle or Hatch and Slack pathway, and C4 pathway. Why this is called C4 pathway? Because the first product of carbon dioxide assimilation is a 4-carbon compound which is known as oxaloacetic acid. So there were very many scientists जिन्होंने ये ऑब्जर्व किया कि कुछ एक प्लांट्स हैं जिनमें फर्स्ट जो कार्बन डाइऑक्साइड एक्सेप्टर है वो RUBP नहीं है, राइबोलोज 1,5 बिसफॉस्फेट, और जो फर्स्ट प्रोडक्ट है कार्बन डाइऑक्साइड एसिमिलेशन का वो 3 कार्बन का कंपाउंड नहीं है, दैट इज़ द 4 कार्बन कंपाउंड. So that leads to discovery of this other pathway of carbon dioxide fixation, that is C4 pathway. So there were scientists like Kortschak Hartt and Burr 1965. उन्होंने फर्स्ट टाइम ये रिपोर्ट किया कि जो CO2 फिक्सेशन होती है शुगर कैन के लीव्स में वो डिफरेंट होती है C3 प्लांट्स में. हाउ दे हैव डन दिस, दे लेबल्ड दिस कार्बन डाइऑक्साइड विद द रेडियो आइसोटोप ऑफ कार्बन, दैट इज कार्बन 14. And they obtained a 4-carbon compound and the aspartate, malate and aspartate as the major labeled product. तो उन्होंने ये ऑब्जर्व किया कि जो 4 कार्बन के कंपाउंड्स थे लाइक malate, that is malic acid or aspartic acid, these two compounds were the major products of carbon dioxide assimilation. जो कार्बन फिक्सेशन के जो मेजर प्रोडक्ट्स थे वो थे malate और aspartate. Then finally जो ये रिपोर्ट किया है that was reported by Hatch and Slack in 1967, और इन दोनों साइंटिस्ट के नाम पे इस पाथवे का नाम भी है Hatch and Slack pathway. They reported that CO2 fixation pathway other than C3 pathway for some tropical grasses. इन्होंने सबसे पहले कुछ एक tropical grasses में देखा कि जो CO2 fixation होती है that is different from द C3 प्लांट्स, और इसमें जो फर्स्ट प्रोडक्ट होता है कार्बन डाइऑक्साइड एसिमिलेशन का वो है 4 कार्बन का कंपाउंड जिसका नाम है oxaloacetic acid. एंड द फर्स्ट टेबल ऑफ CO2 फिक्सेशन वाज 4 कार्बन कंपाउंड, दैट इज oxaloacetic acid. Why this is known as dicarboxylic acid cycle? क्योंकि this is the picture of oxaloacetic acid and it contains 2 carboxylic group. One is this one and other is this one, so that's why this is known as dicarboxylic acid cycle. So this is the first product of carbon dioxide assimilation in case of C4 plants. और जो C4 pathway है ये कुछ एक dicots में भी होती है in addition to monocots, और इसमें जो examples हैं dicots के which show C4 pathway are like common Purslane, the botanical name is Portulaca oleracea, then common saltwort and its botanical name is Salsola, and frosted orache, its botanical name is Atriplex laciniata, then green amaranth and its botanical name is Amaranthus viridis, then Mediterranean saltbush और इसका botanical name है Atriplex halimus. These are the examples of some dicots which show C4 pathway. ये कुछ example है plants के जो C4 pathway show करते हैं, and these are dicots. Mostly जो C4 pathway है ये show करते हैं tropical grasses. Grasses are the monocot plants. The next, there is a difference in the anatomy of leaves in case of C3 and C4 plants. जो C4 plants हैं उनकी leaf की anatomy typically different होती है as compared to C3 plants that we will discuss. A cross section of typical C3 leaves reveals one major cell type that has chloroplast, the mesophyll. जो C3 plants हैं उनमें एक ही तरह के cells present होते हैं leaves में जिसमें क्लोरोप्लास्ट प्रेजेंट होता है और उस टिश्यू को हम कहते हैं मेसोफिल इन केस ऑफ C3 प्लांट्स. जबकि जो C4 प्लांट्स है टिपिकल C4 लीफ हैज़ 2 डिस्टिंक्ट क्लोरोप्लास्ट, इसमें 2 डिफरेंट तरह के क्लोरोप्लास्ट प्रेजेंट होते हैं, 2 डिफरेंट तरह के क्लोरोप्लास्ट कंटेनिंग सेल्स प्रेजेंट होते हैं. वन इज़ द मेसोफिल जो कि नॉर्मली C3 प्लांट्स की तरह है, एंड द सेकेंड वन इज़ बंडल शीथ. Bundle sheath is a structure which encircles the वेस्कुलर बंडल्स, जो वेस्कुलर बंडल्स को एनसर्कल करता है। सो इसमें दो डिफरेंट तरह के सेल्स प्रेजेंट होते हैं विच कंटेन्स क्लोरोप्लास्ट। वन इज़ मीजोफिल और अदर इज़ बंडल शीट सेल। तो इसमें जो क्लोरोप्लास्ट है दोनों में प्रेजेंट होता है एंड दैट वेरीज। और इसकी जो बंडल शीट है दैट शोज क्रैन्स एनाटमी। और इस बंडल शीट को क्रैन्स कहा जाता है जर्मन में, विच मीन्स रिथ। So this is the special feature of C4 plants, that C4 plant leaves shows crans anatomy. Crans means bundle sheath, और bundle sheath compact होती है और ये cover करती है vascular bundles को. So this is the picture which shows the anatomy of C3 and C4 plants. So first picture shows C3 लीव। जो C3 प्लांट्स के लीव्स हैं इसमें सिर्फ और सिर्फ मीजोफिल सेल्स प्रेजेंट होते हैं और इसके अंदर ही क्लोरोप्लास्ट प्रेजेंट होता है। जबकि जो बंडल शीट सेल है दे लैक क्लोरोप्लास्ट। सो इसमें क्लोरोप्लास्ट प्रेजेंट नहीं होता है बंडल शीट के सेल में। सो दिस इज अ राइस प्लांट। राइस प्लांट इज अ C3 प्लांट। देन देयर इज एन एग्जांपल ऑफ C4 प्लांट। इसमें दो तरह के सेल्स होते हैं विच कंटेन क्लोरोप्लास्ट। सो sorghum, this is the example of C4 plant. In C4 plants इसमें मेसोफिल सेल्स प्रेजेंट होते हैं, दीज आर द मेसोफिल सेल एंड दिस इज द बंडल शीट सेल। और जो बंडल शीट सेल्स हैं दे आल्सो कंटेन क्लोरोप्लास्ट, और जो मेसोफिल के सेल्स हैं उनमें भी क्लोरोप्लास्ट प्रेजेंट होता है। और जो इसकी बंडल शीट है इसको कहा जाता है क्रैन्स और रिथ। सो C4 plants show crans anatomy. So this is the major difference in the anatomy of C3 and C4 plants. इसके जो leaves होते हैं, जो C4 plants के leaves हैं, they have two different types of cells, bundle sheath cells and mesophyll cells, and both of these cells contain chloroplasts. So this is the difference. By this picture we can understand how CO2 is fixed in C4 plants. तो दो तरह के सेल्स हैं। पहले जो CO2 है कार्बन डाइऑक्साइड है, ये कंबाइन करता है एक कार्बन डाइऑक्साइड एक्सेप्टर के साथ, वो है C4 प्लांट्स में जो कि मेसोफिल सेल में प्रेजेंट होता है। दिस इज द मेसोफिल सेल ऑफ C4 प्लांट। उसका नाम है PEP, दैट इज फॉस्फोइनोल पायरुवेट। इन अपकमिंग स्लाइड वी विल डिस्कस विद द हेल्प ऑफ पिक्चर दैट। और जो PEP है व्हेन इट कंबाइंस विद CO2, दिस PEP इज अ 3 कार्बन कंपाउंड। जब ये कंबाइन करता है कार्बन डाइऑक्साइड के साथ इट लीड्स टू फॉर्मेशन ऑफ द 4 कार्बन कंपाउंड जिसको कहा जाता है oxaloacetic acid, and it is the first stable compound of the C4 cycle. Next we will discuss in upcoming slide. So this is the wreath जो ये इमेज है ये है wreath की इमेज। नेक्स्ट इन केस ऑफ C4 प्लांट्स, जो C4 प्लांट्स हैं उनमें जो प्राइमरी कार्बन डाइऑक्साइड एक्सेप्टर है वो है एक 3 कार्बन का कंपाउंड जिसका नाम है फॉस्फोइनोल पायरुवेट, और ये प्रेजेंट होता है C4 प्लांट्स के मेसोफिल सेल्स में। और यहाँ पे कार्बोक्सिलेशन होती है, दैट इज एडिशन ऑफ कार्बन डाइऑक्साइड। सो कार्बन डाइऑक्साइड कंबाइंस विद दिस 3 कार्बन कंपाउंड फॉस्फोइनोल पायरुवेट, और ये बना देता है एक 4 कार्बन का कंपाउंड जिसको कहा जाता है oxaloacetic acid. So this leads to formation of a 4 carbon compound जिसका नाम है oxaloacetic acid और dicarboxylic acid. अब जो ये रिएक्शन है इट इज़ कैटलाइज्ड बाय एंजाइम फॉस्फोइनोल पायरुवेट कार्बोक्सिलेज, क्योंकि जो टाइप ऑफ रिएक्शन है दैट इज कार्बोक्सिलेशन, एडिशन ऑफ कार्बन डाइऑक्साइड. सो दिस रिएक्शन इज कैरिड आउट बाय दिस एंजाइम. And this is the picture which shows again the anatomical differences of C3 and C4 plants. जो C3 लीफ है इसमें जो मेसोफिल प्रेजेंट होता है, इसमें मेसोफिल में ही सिर्फ क्लोरोप्लास्ट प्रेजेंट होता है, while जो बंडल शीट है दैट लैक्स क्लोरोप्लास्ट. while in case of C4 plants, जो C4 प्लांट है इनके मेसोफिल सेल्स में as well as bundle sheath cells, both of these cells contain chloroplast. So this is the anatomical difference in the leaves of C3 and C4 plant. जो oxaloacetic acid है it readily equilibrates with other 4 carbon dicarboxylic acid like malic acid and aspartic acid. जो ये oxaloacetic acid है ये immediately it is converted into some other 4 carbon acids like malic acid or aspartic acid, because जो oxaloacetic acid है ये मेसोफिल के सेल्स से बंडल शीट के सेल्स में एंटर नहीं कर सकता है, इट इज़ इम्परमेबल. So a bundle, these mesophyll cells, or we can say the bundle sheath cells, are impermeable to oxaloacetic acid. So that's why it is converted into other 4 carbon dicarboxylic acid like malic acid or aspartic acid. And then the malic acid, जो malic acid बनता है, it is transported to the बंडल शीट सेल. तो क्लोरोप्लास्ट के सेल्स में ये बनता है और ये ट्रांसफर हो जाता है बंडल शीट के सेल में. एंड दिस रिएक्शन of formation of malic acid is carried out by the enzyme malic dehydrogenase. और इस रिएक्शन को कैटलाइज करता है एंजाइम मैलिक dehydrogenase by using a reducing agent NADPH2, that is nicotinamide dinucleotide phosphate reduced form. ये इसकी प्रेजेंस में ये रिएक्शन कैटलाइज होती है. CO2 fixation in case of C4 plants results in formation of this product which is known as oxaloacetic acid, but it is a short-lived product and it is converted into 4 carbon acids like aspartic acid and malate. और जो aspartic acid है, it is produced by the process of transamination by the enzyme aspartate transaminase from this oxaloacetic acid. And after the formation of these 4 carbon acids, ये जो acids हैं, they are transported to the other cell, the second type of cell, that is a cell of bundle sheath of C4 plants. और वहाँ पे ये जो acids हैं, they release carbon dioxide and that process is known as decarboxylation. So this 4 carbon acid is then transported to the chloroplast of bundle sheath cell. Oxaloacetic acid से जब ट्रांसएमिनेशन होती है तो एस्पार्टिक एसिड बनता है या फिर मैलिक एसिड बनता है. और जो मैलिक एसिड है that can enter inside the bundle sheath cell, so that is transported into the bundle sheath cell. And here the malic acid is decarboxylated, that is it will release its carbon dioxide in the presence of nicotinamide dinucleotide phosphate oxidized form. And the enzyme is malic enzyme which will catalyze this reaction. Or after the release of this carbon dioxide, it is converted into the 3 carbon compound which is known as pyruvic acid. और जो carbon dioxide है that is released inside the bundle sheath cell. और जो bundle sheath cells हैं, they contain chloroplast and they behave similar to that of the mesophyll cells of C3 plants. So इनमें enzyme present होता है rubisco. So this is the anatomy that we have already discussed. जो C4 plants हैं, इनकी जो leaves में bundle sheath cells हैं, they bear chloroplast that we can see in this picture. In addition to this, they have second type of cells which are mesophyll cells. They are meant for the process of photosynthesis. So initial जो carbon dioxide fixation है that occurs in mesophyll cell, और इसमें जो carbon dioxide acceptor that is phosphoenol pyruvate converted into the 4 carbon compound which is known as oxaloacetic acid. And after the formation of this compound, it is a short-lived compound. एक convert हो जाता है 4 carbon acids में like aspartic acid and malate, and then that is transported inside this mesophyll cell. और जो ये leaves होते हैं C4 plants के, they show special type of anatomy that is known as Kranz anatomy that we have already discussed. Next are some differences as compared to C3 plants. जो C4 plants होते हैं, उनके leaves में 2 तरह के cells होते हैं. One is mesophyll cell that contain chloroplast, and second is bundle sheath cell that also contain chloroplast. And there is special separation of acids and sugars. So sugar synthesis occur in this bundle sheath cell, and acid formation occur in mesophyll cell. Then next is fixation enzymes. It contains 2 types of enzymes. जो C4 leaves होते हैं, 2 तरह के enzyme present होते हैं. First is pep carboxylase, जो कि mesophyll के cells में present होता है. It helps in carboxylation of phosphoenol pyruvate to oxaloacetic acid. And second type of enzyme which is meant for the synthesis of carbohydrate or glucose, that is rubisco, that is ribulose 1,5-phosphate carboxylase oxygenase, and this enzyme is present in the bundle sheath of C4 plants. और यहाँ पे जो carbon dioxide है that is reduced twice. In this picture we can see here, this is the mesophyll cell. Initially जो carbon dioxide है, atmospheric carbon dioxide that is fixed in the chloroplast of mesophyll cell with the help of enzyme this PEP carboxylase, and the carbon dioxide acceptor is this 3-carbon compound, that is phosphoenol pyruvate. When it combines with this carbon dioxide, it will form the 4-carbon compound that is known as oxaloacetate or oxaloacetic acid. It is short-lived. It is converted into this 4-carbon acid that is known as malate. और ये जो malate है, it can enter inside the bundle sheath cell. ये bundle sheath cell में enter करता है. Here the process of decarboxylation occur. यहाँ पे कार्बन डाइऑक्साइड रिलीज हो जाती है, एंड this malate is converted into pyruvic acid, 3-carbon compound, 1 carbon dioxide is released, and this carbon dioxide is utilized in the Calvin cycle, just similar to that of C3 plants, and then they will synthesize their food in the bundle sheath cells of C4 plants because it contains the enzyme rubisco, that is ribulose 1,5-phosphate carboxylase oxygenase. So this is the brief idea about the C4 pathway. Then after the release of carbon dioxide, there is increased concentration of carbon dioxide in the bundle sheath cells, and these cells contain enzymes which is responsible for the Calvin cycle, that is rubisco, and produces carbohydrate. Then the final step is regeneration of phosphoenol pyruvate. After the formation of pyruvic acid in the bundle sheath cells of C4 plants by the process of decarboxylation, ये जो pyruvic acid है, it moves back to the mesophyll cell. ये mesophyll cell में वापस चला जाता है और वहाँ पे it gets phosphorylated with the help of enzyme pyruvate orthophosphate dikinase, and results in the formation of this phosphoenol pyruvate, which is the carbon dioxide acceptor in case of C4 plants. So this is the picture which shows this whole pathway. Initially this carbon dioxide diffuses inside the mesophyll cell of C4 plants, और यहाँ पे ये water के साथ combine करके बनाता है bicarbonate ion. It combines with this 3-carbon compound, that is phosphoenol pyruvate, and it results in the formation of this 4-carbon compound, that is oxaloacetic acetic acid or oxaloacetate, with the help of an enzyme this PEP carboxylase. This is very very important enzyme. ये enzyme इसलिए important है because जो C4 plants होते हैं, they are more efficient as compared to C3 plants because this enzyme has great affinity for CO2 at a very low concentration of carbon dioxide. Then this oxaloacetic acid is short-lived compound. It is converted into another type of acid which is known as malate. This is again a 4-carbon carbon acid with the help of this reducing agent NADPH. And after that, this malate will diffuses into the bundle sheath cell. This is the bundle sheath cell, और ये जो bundle sheath cell है, यहाँ पे ये जो malic acid है, it gets decarboxylated, means यहाँ पे जो carbon dioxide है ये release हो जाएगा और ये बना देगा एक 3-carbon का compound which is known as pyruvate. और जो ये कार्बन डाइऑक्साइड है, इसकी कंसंट्रेशन यहाँ पे मीन्स बंडल शीट के सेल में इंक्रीज हो जाएगी, एंड दिस carbon dioxide is utilized by the enzyme rubisco as in case of C3 plants, RUBP carboxylase. So this enzyme is present in the bundle sheath cells of C4 plants. Then this enzyme use it in Calvin cycle that results in the formation of sucrose, the stored food material. And this pyruvate is diffuses back into the mesophyll cell. Here it gets phosphorylated with the help of enzyme pyruvate phosphate dikinase, and by using ATP. ATP is a phosphate donor, and it converts back it into phosphoenol pyruvate, which is the acceptor of carbon dioxide in case of C4 plants. So this is all about the C4 cycle or C4 pathway or Hatch and Slack pathway. And this picture again shows the C4 pathway. Initially this atmospheric carbon dioxide diffuses into the mesophyll cell of C4 plants. Here it is converted into the bicarbonate ion. This bicarbonate ion combines with 3-carbon compound, that is phosphoenol pyruvate, and results in the formation of this 4-carbon acid which is known as oxaloacetic acid. 4-carbon acid is formed, that is oxaloacetic acid. Here it is immediately converted into the C4 acids क्योंकि ये जो compound it is short-lived compound. Then that acid diffuses into the bundle sheath cell of C4 plants, और यहाँ पे bundle sheet cell पर this C4 acid undergoes decarboxylation, means the release of carbon dioxide. यहाँ पे carbon dioxide को release करेगा and it is converted into the C3 acid again, that is pyruvic acid. और ये जो carbon dioxide यहाँ पे produce होता है, it is utilized in Calvin cycle. Why? Because in this cell it contains enzyme that is rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase, that helps in the formation of carbohydrate. Then this C3 acid is transported back into the mesophyll cell. Here it undergoes phosphorylation reaction, means addition of phosphate to this C3 acid, and it is converted again into the phosphoenol pyruvate. And by this way, this C4 pathway will continue. So this is all about the C4 pathway. Next, we will discuss about differences between C3 and C4 plants. In case of C3 plants, this carbon dioxide fixation occurs in mesophyll cells only, while in case of C4 plant it occurs in mesophyll as well as bundle sheath cells. CO2 acceptor in case of C3 plant is RUBP, while in case of C4 plant in mesophyll cell it is phosphoenol pyruvate, while in case of bundle sheath cell it is RUBP. Then in C3 plants, first product of CO2 fixation is 3-carbon compound, that is PGA, phosphoglycerate, while in case of C4 plants it is 4-carbon compound, oxaloacetic acid. Then in C3 plants, Cran's anatomy is not present, while in case of C4 plant, Cran's anatomy is present. And in C3 plants, granum is present in mesophyll cells, while in case of C4 plants, granum is present in mesophyll cells and it is absent in the bundle sheath cells. This is a difference in the chloroplast of this C4 plants. Then the chloroplast is normal in case of C3 plants, while in case of C4 plant it's dimorphic. Then optimum temperature for C3 plants are 20 to 25 degrees Celsius, while in case of C4 plant it is 30 to 45 degrees Celsius. So they can grow well in high temperature. Then fixation of CO2 occurs at 50 parts per million concentration of carbon dioxide, while this fixation of CO2 occurs even less than 10 ppm. So I have already told that in C4 plants are highly efficient plants. ये CO2 fixation करते हैं low concentration of carbon dioxide में. So they are less efficient due to higher phosphorespiration. Phosphorespiration की वजह से ये as efficient नहीं होते, while this process is absent or very poor in case of C4 plants. Then RUBP जो enzyme है carboxylase, Ruisco enzyme, it is used for fixation. Here two enzymes, that is PEP carboxylase and RUBP carboxylase are used. Then total ATP used in case of synthesis of one glucose molecule are 18 ATP, while in case of C4 plants it utilizes 30 ATP. So C4 plants requires 5 ATP for the fixation of one carbon dioxide. For the fixation of one carbon dioxide, C4 plants require 5 ATP. So for the formation of one glucose molecule it requires 30 ATP. So it is energy consuming process. Then efficient at low concentration of CO2, while C4 plants are highly efficient as at lower concentration or at higher concentration of the CO2. Then examples of C3 plants are paddy, wheat, potato and some other plants, while in case of C4 plants it includes sugarcane, maize, sorghum, amaranthus, etc. So this is the difference. These are some differences between C3 and C4 plants. The next topic is crassulacean acid metabolism. It occurs in some desert plants and it was first observed in the member of family Crassulaceae. Crassulaceae family के member में सबसे पहले ये mechanism observe किया गया था. So that's why this is known as crassulacean acid metabolism. So this type of CO2 fixation occurs in the member of Crassulaceae, family Crassulaceae. For example, Bryophyllum, Calonche, Sedum and Calcinium. So these are some plants जिसमें कि ये special mechanism observe किया जाता है. It occurs mostly in desert plants. And जो desert plants हैं, to avoid excessive transpiration, इनके जो stomatas होते हैं they are closed during daytime. और daytime में इनके stomata close होते हैं, so the carbon dioxide fixation occurs during night time. And stomata opens during night allowing CO2 to diffuse into the leaves. So रात में ही CO2 को फिक्स किया जाता है. Then here it combines with this carbon dioxide combines with the phosphoenol pyruvate to form this oxaloacetic acid in the presence of enzyme PEP carboxylase. It is similar to that of C4 plants. Then this oxaloacetic acid is then reduced to malic acid, just similar to that of C4 plants with the help of enzyme malic dehydrogenase and NADPH2. So this is similar to that of C4 plants. Here, the malic acid is stored in the vacuoles. As a result of CO2 fixation is known as dark acidification or nocturnal acidification. So रात में ही ये CO2 fixation होता है and then this carbon dioxide combines with PEP and gets converted into the acidic substance that is malic acid. So that's why this is known as dark acidification or nocturnal acidification. So this is the plant Crassula. It is a fleshy desert plant, Crassula. Next, during daytime जो stomata है they are closed और जो malic acid है it undergoes decarboxylation process in the presence of NAD based malic enzyme. So during daytime जो malic acid है इसमें से carbon dioxide release हो जाता है in the presence of malic enzyme, and such depletion of organic acid is known as deacidification. So उस तरह की जो depletion होती है organic acids की like malic acid की that undergoes decarboxylation is known as deacidification. और जो stomata होते हैं they are closed and it increases internally the concentration of CO2. Internally जो concentration है CO2 की increase हो जाती है because जो vacuole है vacuole से malic acid बाहर निकलता है और decarboxylation process शो करता है CO2 release करेगा and results in the formation of 3 carbon acid that is pyruvic acid. And stomata इसमें close होते हैं so that carbon dioxide cannot escape from the leaf, instead it fixed by Calvin cycle. और जो carbon dioxide है वो fix होती है during daytime and it shows Calvin cycle और उसके result से it synthesizes food material. So then the pyruvic acid is formed. जो pyruvic acid है वो form होता है after decarboxylation of malic acid and may be converted back into phosphoenol pyruvate by the pyruvate orthophosphate diekinase enzyme and magnesium, and is just similar to that of C4 plants. So we can say जो crassulation acid metabolism है ये जो plants हैं during night time they show C4 pathway and during daytime they show C3 pathway. So this release of CO2 is accepted by RUBP, that is ribose 1,5-bisphosphate. This is a 5-carbon compound and here the enzyme is Robisco and is fixed in C3 cycle, just similar to that of C3 plants. So this is the picture which shows this process during night time, dark time. जो stomata है वो open होते हैं और जो carbon dioxide है atmospheric carbon dioxide that gets diffused inside the mesophyll cell और यहाँ पे it combines with water to form bicarbonate ion, and here it combines with this 3-carbon compound that is phosphoenol pyruvate और ये जो phosphoenol pyruvate है ये combine करता है this bicarbonate ion के साथ in the presence of enzyme phosphoenol carboxylase and results in formation of this 4-carbon acid that is known as oxaloacetic acid. Here this acid is short-lived and it is converted into the another 4-carbon acid which is known as malate in the presence of malic dehydrogenase enzyme. And after the formation of this malate is stored inside the vacuole. जो malic acid है during night time this is stored inside the vacuole. So malic acid को store किया जाता है during night time in the vacuole of this mesophyll cell of Crassula plant. During daytime what happens, जो stomata है वो close हो जाता है. So ना ही यहाँ पे carbon dioxide अंदर enter कर सकता है और ना ही यहाँ से carbon dioxide बाहर escape करता है. Then during daytime जो malic acid है it comes out of the vacuole और जो malate है it undergoes decarboxylation process. Decarboxylation means release of CO2. With the release of CO2, ये जो CO2 यहाँ से release करता है this results in the formation of this pyruvate which is a 3-carbon acid. और ये जो 3 कार्बन एसिड है दिस पायरुवेट आइदर इट इज कन्वर्टेड इनटू स्टार्च और आइदर इट इज कन्वर्टेड बैक टू phosphoenol pyruvate. That is the regeneration step. In this picture we can see here. So from starch triose phosphate is formed and then it is again converted into phosphoenol pyruvate, which is the acceptor of this carbon dioxide. While this CO2 is utilized in the Kelvin cycle. जो CO2 है ये Kelvin cycle में use किया जाता है for the formation of glucose, that is the reserve food material. So this is all about this cressolation acid metabolism. So we can say that during night time or dark time this shows C4 pathway, while during daytime it shows C3 pathway. So this is all about the cressolation acid metabolism. This is again same picture. During night CO2 is fixed and it is converted into this oxaloacetic acid with the combination of phosphoenol pyruvate. Then it is converted to malate. It is stored in the vacuole. During daytime this malic acid comes out of the vacuole, or जो malic acid it undergoes decarboxylation reaction. During daytime stomata are closed. Then this carbon dioxide is fixed in Kelvin cycle, while this pyruvate is converted back into starch. By the breakdown of starch, it results in the formation of, from this picture we can understand, triose phosphate, 3 carbon for this sugar. Then it is converted back to phosphoenol pyruvate, and this is the carbon dioxide acceptor, 3 carbon compound. So this is the process how it is regenerated. This is all about this cressolation acid metabolism. Next we will discuss about photosynthetic plasticity. ये एक प्रॉपर्टी है प्लांट्स की टू शिफ्ट बिटवीन C3 एंड कैम मेटाबॉलिज्म, और ऐसे प्लांट्स को कॉमनली कहा जाता है फैकल्टेटिव कैम प्लांट्स. So it is the property of plants which can switch between C3 and CAM mode of photosynthesis, और इनको commonly कहा जाता है facultative CAM plants. There is a one plant which is known as Mesembryanthemum crystallinum. This is commonly known as ice plant. ये एक annual plant है, वो switch करता है this photosynthetic modes को, इस photosynthetic mode को in response to water and salt stress. जैसे water और salt stress होता है वैसे ये switch करता है this mode of photosynthesis. ये अपनी लाइफ को स्टार्ट करता है बाय यूजिंग C3 पाथवे. जो आइस प्लांट है इनिशियली इट शो C3 पाथवे बट स्विचेस टू कैम पाथवे लेटर ड्यूरिंग फ्लावरिंग स्टेज, जब इसमें फ्लावर्स बनते हैं और अंडर स्ट्रेस कंडीशंस. जैसे ही सॉल्ट या वाटर स्ट्रेस होता है इस प्लांट में ड्यूरिंग लेटर स्टेजेस ऑफ लाइफ एंड ड्यूरिंग फ्लावरिंग, ये जो प्लांट्स हैं ये शो करता है कैम मेटाबॉलिज्म. And this flexibility allows species to utilize C3 pathway. ये जो flexibility है ये plant grow करता है C3 pathway से in the initial stages, और जब water की availability अच्छी होती है and it increases its biomass by showing the higher rate of photosynthesis. So जब conditions normal होती है तो ये plant use करता है C3 pathway of photosynthesis and increases their stress tolerance during the drier period through CAM pathway. जब conditions tough होती है तो ये stress tolerate करता है by using the second pathway which is known as CAM pathway. So this is an adaptive mechanism to avoid the stress conditions. So this is all about today's discussion. Hope you will understand this. If you have any questions, queries and any suggestions, you can give in the comment section. Thank you. Have a great day.